Hello everyone, this is our first biology review video of the year. It's not going to be very exciting. It's just me going over the review questions with you, but this should be helpful if you have some questions regarding to the review questions. First thing, you want to make sure that you have your name, your full name, spelled out very clearly on tomorrow's test. You want to have your section number. It's either BM4 or BM7. If you have fourth period every single day, you're BM4. If you don't have first period every single day, fourth period every single day, then you are going to be BM7. Your student number is on your name card the first day of school. If you don't remember what it is, feel free to come see me. So the first question is, what is the difference between quantitative and qualitative data? So let me give you an example. A quantitative data is always something that you can measure or count. So for example, one apple, that's a quantitative data. If you have 30 inches of a piece of wood, that would also be a quantitative data. So anytime you see a number, or if you're thinking about something that you can count or measure, or let's say 30 liters of water, this is all quantitative data. Examples of qualitative data, basically everything else. When you can describe the outside appearance of something, when you can describe the color, the size, and all that. Anything that you cannot measure directly. So let's say a green door, that'll be ugly. That's a qualitative data. If you say a soft couch, that's a qualitative data. A tall tree, that's a qualitative data, okay? So you want to make sure that you are able to come up with an example of quantitative data and qualitative data on your own, and make sure you don't get the two mixed up. If you just look at the word quantity, you know what a quantity is. It's something that you can measure, right? Second question, what are the steps of scientific method? We went over all of this when we did our termite lab. But basically, here are the steps. You first have to make an observation, and then you ask a question. So let's say for our termite, we made the observation that termites can follow the ink of the big pen sometimes. So your scientific question is, can a termite follow a circle drawn using the blue big pen? Your hypothesis is termites can follow a circle drawn using a blue big pen. Then you conduct the experiment, which is what we did yesterday. We collected the data, which is when you did the measurement, well, when you look at the timer and then observe the behavior of the termites. So you really do observation way ahead of time, as well as when you're collecting the data. But I do want you to remember that you have to do an observation before you ask a question or provide a hypothesis. And then you collect the data, we did that. And then you do a data analysis, which is when you counted the number of seconds when a termite was on the blue line. That's your data analysis. You wrote down all those numbers, whether the termite is on the line, not on the line, walking away from the line, sitting still. Then you have to look at that data and kind of analyze it a little bit. Yesterday's was pretty easy. We're just counting how many seconds the termite stayed on the line. Lastly, you make a conclusion. Your conclusion should be either a termite would follow a circle drawn using a big pen, or a termite would not follow a circle drawn using a big pen, okay? Just real quick, you want to make sure that you know a scientific question is always a question, right? If you ask, well, it has to be something science related, so you could ask, why is grass green? That's a scientific question. Hypothesis is kind of a statement that's your best attempt in answering this question. So you might do some research, you might look online, why is it that grass is green? So maybe you find out grass is green because the chloroplasts in the plant cells are green, okay? So that statement right there that you decide might be an answer for your scientific question is going to be your hypothesis. The next one is, what are the characteristics of life? For this one, I guess just remember it. I don't have a good way for you to memorize it, but you should have a general sense that, let's say, moving is not a characteristic of life, but all life has cells and all these. Number 4, this is really important because you're going to be asked to write out the independent variable, dependent variable, experimental group, and control group, and it's not going to be a multiple choice question. So you want to make sure that you know how to do this type of question. So I'm going to give you A as an example. I'm going to walk you through the independent variable and all that. And B is going to be your homework. So you could potentially do it as a practice. And if you want to send me your answers and have me check it over for you, you could do that. Okay? So A, first off, is a joke. Don't eat Tide Pods. And you do not want to actually test this experiment. It's just a joke. But if this were to be real, you want to test the effect of people eating Tide Pods on the number of emergency room visits. So this is kind of what you're trying to test, right? Your scientific question would be, what is the effect? All right, let me just write it down. Scientific question. So let's say you're wondering about this thing, but you want to make it more scientific. So you start with asking a question, scientific question. What is the effect of people eating Tide Pods on the number of their emergency room visits? And then you make a hypothesis. So remember, a hypothesis is a statement that's your best answer to the question. So your hypothesis could be something that makes sense to you. Let's say people who eat more Tide Pods have increased number of emergency room visits. Okay, so that's your hypothesis. Basically, you're asking a question and then you're answering this question to your best ability. The next one is independent variable. Independent variable is always something, it's the cause, okay? I'm going to highlight this. It's the cause of something that happens. Right. So your dependent variable is the effect of something that happens. So in this case, if you look at your hypothesis, people who eat more Tide Pods have increased number of emergency room visits. What is the cause? What is the effect? One way to do this is to change this sentence into an if-then statement. If you want to change, then you can see the cause and effect a lot more clearly. So if you do that, it's going to be changed into if people eat more Tide Pods, he or she would have an increased number of emergency room visits, right? Oh wait, I missed it then. If people eat more Tide Pods, then he will have more emergency room visits. So our cause is Tide Pod. So you really want to make sure, well, more like the number of Tide Pods, the Tide Pod. So you really want to make sure that you're not saying more Tide Pods. So this is wrong, okay? More Tide Pod is wrong. Don't do it. That's wrong. The reason why more Tide Pod is wrong is when you're talking about a variable, it's something that can go both ways. So it can go larger, no change, smaller. If you say more Tide Pod, it's no longer a variable. It's already something that happened. It's more Tide Pod. But if you were to say Tide Pod or the number of Tide Pod or whether people eat Tide Pod, that's something that has a change. So the number of Tide Pod could be 0 or more Tide Pod, 0 or more Tide Pod. Whether people eat Tide Pod or not, you have people who don't eat Tide Pod, you have people who do eat Tide Pod. So there's a direction that you can change the variable. The next one is your dependent variable is the effect. The effect is the number of emergency room visits. So you would not say what? You would not say increased number of emergency room visits. This is wrong, okay? Don't do it, because the increased number of emergency room visit is no longer something that varies. It's already increased. It doesn't change anymore. But if you say the number of emergency room visits, it can go less number, more number, okay? Now, the second example, you can use it as a practice, and if you want to send me the answers, you could. Number 5, know the different parts of the microscope. I can't help you memorize it. Just memorize it to the best of your ability. I'm going to post this Kahoot link onto our Veracross calendar, so you can use it as a practice if you would like to do that at home or with a friend or with your parents even. See how good you are at remembering the parts of the microscope. Number 6, we already went over this in class, how to use the microscope. That's basically the video that we watched in class, which is on the PowerPoint that's posted in our Google Drive folder. When you first look at a sample, which lens do you start with? You always start with the lowest magnification, okay? How to carry the microscope, you use one hand on the arm of the microscope and the other hand on the base of the microscope. What is the purpose of the cover slip? Two things. Number 1, you're protecting the lens from the specimen, and you're protecting the specimen from the lens, okay? Number 2, you are flattening the specimen and keeping the specimen in place. So you don't have to write those exact words, but that's the general idea. What is the reason for not using the coarse focus knob when we're using high objective lens? Because we do not want the high objective lens to squeeze onto the specimen itself or squeeze onto the stage, because our high objective lens is very long, and if you use the coarse focus knob, it's going to change the height of the stage too much that it might squeeze the lens. All right, number 7, a metric system. Let's start with the basic units. Time is second, length is meter, volume is liter, mass is gram. So mass is kind of weight of something. Temperature is Celsius, degree Celsius. Whoop. There we go. So you have to be able to convert unit using those prefixes. So you can add any of these prefixes in front of any of these basic units. So let's just try an example real quick. Let's do it right here. So what you also want to remember is these different steps, right? You start with the middle. Every step you take, that's a difference of tenth. Every step you take this way is also a difference of tenth. So a deca, let's say a decaliter. A decaliter is 10 times larger than a liter, a regular liter. A hectoliter, you're jumping 10 times 10. So a hectoliter is 100 times bigger than a regular liter. If you have a deciliter, it's 10 times smaller than a regular liter, okay? So the difference in each step is always 10, but if you're jumping 2 steps, you're using 10 times 10. If you're jumping 3 steps, that would be 10 times 10 times 10. So let's try that real quick. Let's say we're doing millisecond. 20 milliseconds equal to how many seconds? So we're going from a smaller unit right here, a smaller unit to a bigger unit. We're jumping 3 steps, 1, 2, 3. So you know the difference between a millisecond and a second is 10 times 10 times 10, which is 1,000 times. Since millisecond is smaller than a regular second, it's 1,000 times smaller, you want to move your decimal to the left. So you start with the decimal right here, right? This is where your decimal is supposed to be at the beginning. You move it to the left 3 times. 1, 2, 3. So if we do it on here, 1, 2, 3. And then if you enter all the zeros that's supposed to be there, it is 0.02, right? 0.02 second equals to 20 milliseconds. Okay. Another example could be, let's do kilogram. Let's do 3 kilogram equals to how many decigram, even though nobody uses that. So if you look on here, from kilo to deci, 1, 2, 3, 4. The difference between kilogram and decigram is 10 times 10 times 10 times 10 equals to 10,000. Okay, since we're going from a larger unit, kilogram, to a smaller unit, decigram, right here, you want to move the decimal to the right. And since we're doing 10,000, you're moving it to the right 4 times. 1, 2, 3, 4, right? We're jumping 4 times. So your decimal initially starts right here, and you move it 1, 2, 3, 4. And now your answer is 3,000,000. 30,000 decigram, okay? I'm going to post an answer key of the metric conversion worksheet that we worked on in class. If you want to use that as a practice, you can look at the blank worksheet and try to work on it, and then use the answer key to make sure that you know how to do these type of questions. All right, we're almost done. Last one. Where do you need to look at when you are measuring liquid with the graduated cylinder? You want to look at the bottom of the meniscus. So, I don't think I can draw. So if you're looking at a graduated cylinder, for example, right here, okay, you have a liquid. Usually water is a curve that goes under like this. When you're measuring the liquid, you want to look at the bottom of the meniscus, okay? So if this is the curve of the liquid that you see within the graduated cylinder, you want to look at the bottom right here and see what measurement is this, okay? So on the test, you might be asked to read the measurement of a graduated cylinder. I don't have internet right now, so I can't give you an example, but if you want to look up a picture and try reading the measurement of a liquid, and you can send me an email and ask me if you got it right or not. I think you're ready for tomorrow's test. You're going to do great.